And good afternoon, everyone. Welcome to Research Grand Rounds. I'm Dr. Kim Williams, Chair of Medicine, for those of you who are online. And we are so pleased today to have a wonderful discussion of sex-specific environmental liver disease. And hopefully we will all gain some understanding about it and with the subtitle, She Matters Too. And it's going to be presented to us by Banrida Wahlang, who is one of our PhDs in pharmacology, toxicology, and the Department of Medicine. She's has a wonderful background starting in India, where she did her bachelor's degree and a Master of Science in pharmaceuticals or pharmaceutics at the National Institute of Pharmaceutical Education. She then came to University of Louisville and did another Master's of Science, two master's degrees, okay, in pharmacology and toxicology, and then she did her PhD. And so that was a while back. She did several years in postdoctoral positions, became an instructor in the Department of Medicine in 2001, and she's been an assistant professor since 2002. When you look through her CV, it's double-sided and massive number of publications, all of them sharing insights with the world and changing the field as we know it. So thank you so much, Dr. Wahlang. We're so happy that you're here, and we're looking forward to learning more about sex-specific environmental liver disease.
All right. Well, thank you so much, Dr. Williams. I'm really happy to be here today. Before I start, I just want to reveal something. So I cheated. No, that shouldn't be there. Okay, so I cheated. I picked this day for the Research Grand Rounds because yesterday I was supposed to give a talk to the Division of Endocrinology, and I thought, why not kill two birds with one stone? So I'm hoping today that none of you all had listened to my talk yesterday because they are going to be somewhat similar, but there's some new data that I had put in here that are new that I didn't share yesterday. So yesterday's talk was called Battle of the Sexes. And everybody was excited to watch the movie, but I actually told them about my research for an entire hour, and I intend to do the same today too. So really happy to be here, and let's talk about our research. So first of all, there are no relevant disclosures regarding this research. I do want to acknowledge all the different centers and federal agencies that I work with to do this work. All right, so let's talk about environmental liver disease. So what comes to mind when we talk about or we think about environment, right, are chemicals. So, chemical environments, the chemicals that interfere with any apart aspect of the liver, metabolic dysfunction, or even the endocrine system as a whole, because the endocrine system plays an important role in maintaining energy metabolism in our body. But the chemical environment is not just it. There's so much more to it than just chemicals and chemical-induced liver disease. So the physical environment, for instance, where we live, how much cell phone use do we, how much do we have our cell phones in our pockets, or for instance, how much of the laptop do we use? There's also biological environment. For example, sex is a major part of that, but it's also other biological elements throughout the lifespan of a person, such as pregnancy or lactation or other pre-existing health conditions. But there's also sociocultural environment. And I think this plays a really, really big role in terms of shaping or influencing liver disease or even metabolic dysfunction in general, because, for instance, right, social traits influence, where do we eat, how do we eat, do we go to the gym daily, do we take a Zumba class, or do we bathe, right? So really, social cultural environment is also an important element of that. So to make things more complicated, these different environments also tend to interact with one another to influence disease outcomes. All right, so why does environmental liver disease matters? So I was looking up on Google and trying to find the most recent global prevalence of liver disease, particularly metabolic dysfunction associated liver disease. And this map popped up and I was looking at it. One thing that really came to mind for me was that if you look at the prevalence, the countries or the regions in the globe that steatotic liver disease is more prevalent, it actually is not specifically in the States or in North America, or even like in Europe. So this idea that, Western diets, for instance, or the Western lifestyle is the major cause of these steatotic liver disease is really not at play here, because there seem to be so many other factors. And one of the factors could be the demographic shifts today in terms of our food consumption. So there's more processed food, more high calorie food that's available to these regions like the Middle East or even the Sub-Saharan African region, and people are consuming more of those foods. But with that, there also comes chemical exposure, because we now know that chemicals are everywhere. For instance, PFAS are there in the food packaging of McDonald's, for instance. So that's why it matters to study environmental liver disease. And also, another important point is that the liver is the most common target organ for chemical toxicity. And there's been so many publications that have documented chemical exposure with liver disease. So it's really not, this field has been there for a while, and it's really not that new anymore. And we know that chemical-induced liver injury is an important area to study. So with that in mind, our research objectives of my group is to look at sex differences in environmental liver and metabolic health. We're interested in the different endpoints of the metabolic syndrome, steatotic liver disease, and to an extent, we've started dabbling with cholestatic liver disease. Our environmental contaminants of interest are persistent organic pollutants and volatile organic compounds. So I know that the cardiovascular group, as well as Environment Institute, really work a lot more on these VOCs. So today I'll be talking a lot more on these persistent organic pollutants that I passionately call them POPs. They're one of my favorite kind of chemicals. And so what are the objective really of our group? So we really want to define the sex-specific mechanism of action of these environmental chemicals in order to be able to identify at-risk populations. And most importantly, how can we identify sex-specific intervention strategies? And so we adopt a research translational approach in our work. We usually look at observations from group and population studies, take this information to the lab, and then we work with our mouse models that I'm gonna show you here today. And I would like to use that pointer. All right. So steatotic liver disease is one of the manifestations, actually the liver manifestation of the metabolic syndrome and metabolic dysfunction. And for most of you that know it well, this progression from a normal liver to a fatty liver, and then if it's accompanied by inflammation, it becomes to hepatitis, and eventually could proceed to cirrhosis and hepatocellular carcinoma. And in terms of sex-specific liver disease prevalence, so with metabolic dysfunction associated steatotic liver disease, or MASLD, it's known to be more prevalent in males and in postmenopausal females, and this hints at the protective effect of high levels of estrogen in premenopausal females. When it comes to alcohol-associated liver disease, while the prevalence in the past is thought to be more in males, a lot more research have been done in the last 5 years, for instance, that have shown that, with narrowing that knowledge gap on sex differences, that females, it actually is more prevalent in females. And what about toxicant-associated steatotic liver disease, which is our disease of interest? So we don't know much about it. So that's why our research, is highly important. So sex and gender and environmental health can help in explaining some of this disproportionate impact on men and women's health. And we have to remember that this is important because biological processes through the lifespan, as we're exposed to these chemicals through the, in our exposome, if you will, they're different in males and females, and they're confounded by a number of factors like pregnancy and so on. And so from a biological perspective, so what are the factors that influence sex differences? Well, there are multiple standpoints, but I usually like to group them into 4. So one is from a genetic standpoint, right? Differences in XY versus XX chromosome. For instance, the SRY expression in males lead to these gonadal differences, such as, hormonal activational effects, hormonal organizational effects that then affect phenotypic characteristics. Whereas there are also non-gonadal effects, such as the unequal expression of X and Y genes, that could also lead to these sex chromosome effects. And there are also other factors, such as hormone levels between males and females, that play a role, as well as reproductive elements. And so how does sex and gender matter in environmental health? So we talk about sex, but why does gender also play an important role? So this is my classic example. So when you think about a chemical exposure, let's say through dermal absorption. So if you think about, dermal absorption through the skin and skin thickness, that's usually different in our males and females, with males tending to have thicker skin, and that could impact or affect the amount of chemical that's being absorbed into the body and subsequently the dose. But what about gender? How does gender impact, chemical exposure and the amount we're exposed to? Well, it does impact a great deal, because gender is defined by, our identity, our social cultural identity of who we are, and it dictates a lot of our lives. So for instance, women in general tend to use more cosmetic products, more hair products than men, and those could be a source of chemical exposure that, men may not necessarily be exposed to, and that could play an important role. So just an integration of sex and gender and its importance in environmental health research. All right, so I work in the GI division, so I always have to go above and beyond to convince my colleagues sometimes about why sex differences are important in liver disease. So I just want to bring out this nice example here about, sex disparities in liver transplant. So in terms of the leading cause for liver transplant, that's very different in men and women. In women, it's usually metabolic, usually NASH or alcoholic steatohepatitis, whereas in males, it's usually ALD or alcohol-associated liver disease. But sex disparities in liver transplant have been widely known, but they're very poorly understood. So we don't understand why that happens. Women tend to be 30% less likely to have a liver transplant, but they also happen to be more likely to die while waiting for an organ donor than men. And so some of the reasons that have been theorized or speculated in the past few years have been that perhaps there's the way that we define who is eligible for a transplant. So one of them is this MELT score, which is a score that defines the severity of the liver disease sickness, measured by a 3-month period. And usually with this MELT score would measure takes into account, let's say, blood creatinine levels. And that puts women at a disadvantage because women tend to have less muscle mass and subsequently impacts their creatinine levels. And therefore, they tend to have a lower MELT score that puts them at the somewhat bottom of the list than men. But there's also the other factors, like I had mentioned, how men are more at risk for liver disease than women. And so they tend to have more, incidence of hepatocellular carcinoma. And if you have liver cancer, you get more extra points for your MELT score. And so that again puts them at an advantage, whereas women are left behind. So there are some ways that physicians have come about this to kind of perhaps, allocate more points to women in terms of this MELT score. But this is just an example to show you of why sex differences are important, not just in our research, but also in our healthcare system, and recognizing that. All right, so shifting gears a little bit, let's talk about persistent organic pollutants. So these are forever chemicals. And unfortunately, they were all man-made, and they were produced intentionally for industrial processes, or even sometimes they were released unintentionally through, during industrial operations or industrial manufacturing of these chemicals. And I want to show you this nice little map here of the story of, the lifetime of how these chemicals have been recognized to be bad, but still continue to this day to be a problem. So this was a list of chemicals that were listed under the Stockholm Convention when a bunch of countries came together and met in Europe to talk about how can we stop these bad chemicals that these industries have been manufacturing that we've been using. How can we stop them from being used anymore, because they're becoming a problem, not just to human health, but to the ecosystem in general. And so they met, and in 2004, they put a bunch of chemicals such as organochlorine pesticides and PCBs, which are polychlorinated biphenyls, or that I will talk more about today. So they put those chemicals in this list called Dirty Dozen. But, that didn't solve anything, because industries come back with other chemicals like PFAS chemicals, for instance, right? So in 2009, 2010, they listed PFOA and PFOS, which is one of the major PFAS chemicals in this list. But the companies just took that chemical, modify the structural moiety a little bit and produce a new chemical that's not regulated, and continue to go on with this vicious cycle, if you will. And so it never really got resolved. And now we're in 2024, and forever chemicals is still a big concern, and it's all over the news. And a problem with these is that they are resistant to degradation. They're highly fluorinated compounds. They tend to bioaccumulate in the adipose tissue of living organisms, so they eventually amplify through the food chain. Another problem with this is because we know that climate change is now a big issue. And these chemicals have a tendency to grass hop. So what do? They could, from low latitudes where they're present, they could evaporate and travel across the globe and go and deposit at at regions of high latitudes. So another classic example for this is DDT. So DDT is not used in the United States for a long time now, for decades, but as far as I remember, about 10 years ago, it was still used in India and some parts of China even. And so those levels of DDT still persist in those areas. And now with changing temperatures globally, there's a tendency of the potential that these compounds could migrate or grass hop to other regions. So it is still a very big environmental concern. And another thing to remember about these POPs, or persistent organic pollutants, is that they occur in our environment as mixtures. So they're often detected as combinations of chemicals in our soil, water, and air samples. So if you were to catch a fish from the Ohio River and send the fish tissue to the lab to look at what kind of chemicals are there, you'll always find PCBs, you'll always find organic chlorine pesticides, and PFAS in that fish tissue. And I know this because I just saw those numbers a couple of days ago from some fish that was caught from the Ohio River and a month ago. All right, so why do I showed you why we care about sex differences. So are there any sex-specific environmental evidence, that show that perhaps these effects or impact of these chemicals are different in our men and women? And so I had the opportunity to work with this cohort study from Anniston, Alabama. So Anniston is a small town in Alabama that was home to Monsanto Corporation that used to manufacture PCB back in the day. It also is a home to multiple military bases, and we now know that military chemical exposure is also a big health concern. And it also is home to several other manufacturing plants. It was a highly industrialized region, for example, just like Rubber Town in west from here. And so people were also exposed to multiple organic chlorine pesticides. So we were able to work with this cohort that had an initial and a follow-up population. And so we were we first started out by saying, okay, let's look at the levels of these compounds and see if they're different in our males and our females. So we started off with PCBs. And you can see here in both our, phase 1, which is our initial cohort, as well as our follow-up phase 2 cohort, females tend to have higher levels of PCBs than males, so circulating levels. So we saw sex differences in our circulating chemical levels, and we decided to go ahead and look at disease biomarker associations. So I'm going to show you a couple of figures here. So we decided to look at this oxychlordane, which is a metabolite of the organic chlorine pesticide called chlordane, and we did some sex stratified association analysis, and we found that in our females there were positive correlation between blood oxychlordane levels with M65, which is a liver cell death biomarker, and we saw the same thing with interleukin-6 or IL-6, which is a pro-inflammatory marker. But we saw the opposite with our males. So this kind of made us really excited and said, okay, maybe we should really pursue sex differences and see if even disease etiology or disease progression is different based on sex. So another population evidence that I want to show you is this study that we did in collaboration with the UofL Superfund Research Center and Green Heart, where we look at the HEAL study. Many of you here have heard about HEAL. Some of you are investigators in that study. So we decided to look at liver disease biomarker associations with urinary VOC metabolite levels. And we decided to stratify it by sex again. So we had initially published one study, we looked at everybody and we saw these associations between these metabolite levels with liver disease biomarkers only in smokers. And we decided to see, okay, is it really, what's driving that, which sex is driving that difference? Is it males or females? And surprisingly, when we did that, what we saw was that it was only in our females that we actually saw those positive associations. And we saw these associations between 8-VOC metabolites and alkaline 4-phosphatase, but we didn't really see any of those associations in our males. And it's also more pronounced if they were female smokers. So our conclusion was that, females seem to be more at risk that have higher VOC levels seem to be at risk for liver disease than our males. So again, another beautiful evidence of sex-specific associations. And so the next thing we did, so before I go to our animal study, I just want to tell you also how we assess these toxicology endpoints in our lab. So generally back in the day, when it comes to diabetes and obesity, it's been thought that energy intake is not equal to energy outtake or output, and that's why you're having these metabolic complications. And so they're driven by nutritional habits and genetics. But now we know that it's more complicated than that. So we know that it involves a lot of organs. It involves the endocrine system, involves the gut. So it's important to look at multi-organ crosstalk, organ-organ interactions when we look at these factors. So, and we also know that it's not just this energy intake equal energy outtake that matters, but also our lifestyle, right? So what kind of energy are we intaking, right? What kind of fat are we eating? What kind of drinks are we drinking? And also what the environment around us is doing to our bodies. So what are the chemicals that we're exposed to? And also environmental stressors play an important role. And all that could contribute to toxicities to different organs simultaneously that could impact these metabolic dysfunctions. And so with that in mind, one of our, how we approached one of our studies was to look at how these environmentally relevant mixtures of POPs or forever chemicals, because I said that these chemicals are often found in mixtures. And so we've taken the mixture route now. How do they impact multi-organ system, for instance, endocrine gut liver axis, and how is that different in males and females? And we hope to do this to identify mechanisms and hopefully potential intervention strategies. And so I'm going to show you a little bit about our mouse exposure studies and what we found. So we did this one study where we had male and female mice that had a Western diet that had 40% fat or a low-fat diet, and they were exposed to a mixture of POPs, namely chlordane, which is a non-dioxin-like chemical, and PCB126, which is a dioxin-like chemical. And they were exposed and simultaneously fed a high-fat diet for the 12-week period, and then eventually euthanized, and we measured toxicity indices. So what did we see? So the first thing we looked at was blood glucose levels, and this was done in live mice towards the end of the study. And we look at males and females, and if you can look here, our greens are our males, our purple are our females. And if you look at the area on the curve, you can see that there was a significant difference in the amount of blood glucose that was taken up when we look at our, our exposed males versus their sex-specific controls. We never saw that in the females. So this somewhat aligned with that common knowledge that, for instance, type 2 diabetes is more prevalent in males, and males are more susceptible to glucose intolerance. And we also look at physical activity in our animals. And surprisingly, so we put these animals in these metabolic cages that could measure their movement, their food consumption, and so on. And surprisingly, we saw that only our female exposed animals tend to have a lower activity. So they seem to be just hanging out in the cages and really, not as active as their sex match controls. So we were really excited to see this phenotype. And we went ahead and looked at other, at the liver. So we did a liver histology analysis. So we looked at H&E staining and all-red staining. And what we actually saw here was that there were obvious sex differences in the amount of hepatic lipid accumulation with this, Western diet feeding in our Black 6 mice. And we can see that in both our male controls and male exposed, there was this diffused macro and microvesicular steatosis. But if you look at our females, they tend to have lesser amounts or lesser frequency of lipid droplets, and it appeared to be mostly macrovesicular steatosis, and it was more zonal rather than diffuse. So we saw that and we were like, okay, let's measure our hepatic lipid levels and see if there's any difference or it's just sex differences. So we measured our hepatic triglycerides and cholesterols, and we were really excited to see that, yeah, we saw sex differences, we kind of anticipated that, but we saw that in our females with the chemical exposure, there was this slight increase, but still significant increase in our hepatic lipid levels, both triglycerides and cholesterol, suggesting that perhaps in our females, with the chemical exposure, they may be more susceptible to hepatic lipid accumulation with Western diet feeding. And so we wanted to know what was driving this, right? So there were other data that I'm not showing you, but I wanted to show you kind of a big picture. So we wanted to see what are some of the mechanisms that are driving this steatosis in our female mice. So initially, we just look at our hepatic receptor activation. So POPs are xenobiotics. Just like drugs, they can go and activate xenobiotic receptors, such as the aryl hydrocarbon receptor over here and the constitutive androstenedione receptor, or CAR. And so we sent our livers to the proteomics core with Dr. Mike Merchant, and he, sent us back the results and said, okay, these are the number of proteins that were upregulated or downregulated in your mice. So I'm going to show you just a little bit. So our CYP1A1 was upregulated in both our exposed males and females, and it is the target for the aryl hydrocarbon receptor, indicating that with exposure, the AHR was activated. Similarly, CYP2B10 is a target for the constitutive androstenedione receptor, and it was also activated in both our males and females with exposure. CYP3A11, which is a PXR or a pregnant xenobiotic receptor target, was not upregulated, so suggesting that this receptor was not affected. But what was really interesting to us was when we look at our sex exposure interactions, we saw that fatty acid synthase was upregulated. So we went ahead, or rather my undergraduate student went ahead and did some Western blot analysis and confirmed that, CYP1A1 upregulation in our males and female exposed. And what was really neat was that we actually saw that fatty acid synthase was upregulated in our female exposed mice, which may perhaps be some of the answers to that increase in hepatic steatosis. So we asked ourselves the questions, are females more susceptible to steatosis? So luckily at this time, we just got a DoD grant that got funded looking at diopsin-like mixtures. Dioxins are a major, chemical of concern for military. They were components of Agent Orange that was used as a herbicide during the Vietnam War. And so we look at low levels of dioxin exposure. So these chemicals here are, TCDD is a diopsin, and these are diopsin-like chemicals. And we exposed both the males and female mice for 2 weeks to this mixture. And we also found the exact same thing. If we look at hepatic lipids, hepatic cholesterol and triglyceride, they were increased upregulated in our females with exposure. So what was driving this normal liver to steatotic liver, even in the absence of a Western diet feeding? And so my graduate student, Esther, went ahead and ran a bunch of PCRs looking at lipid metabolism genes, and she had some exciting results to show me. We look at it, and it looks like all the enzymes involved in de novo lipogenesis were upregulated with exposure, only in our female mice, but not our males. And so we asked ourselves this question, so how is this happening, and if it could be a sex-specific AHR mechanism? So for those of you that really don't know much about receptor biology, for instance, the aryl hydrocarbon receptor, when it's not activated by a xenobiotic or like by a diopsin, if you will, it just hangs out in the cytosol, and it's usually held together by a number of scaffolding proteins like HSP90. So when it gets activated by a ligand or a drug or a contaminant like diopsin, it gets released separates from its scaffolding proteins and then translocates to the nucleus and does its job as a transcription factor. But another function of the AHR, when it's sitting in the cytosol, it also can have the ability to sequester certain proteins. So one of the proteins that it could do so is the SRABP1, or sterol regulatory element binding protein 1, which is transcription factor for a number of lipogenic genes. And so what happened is that we theorized that in our exposure studies, the diopsins are coming in, activating the AHR, it moves to the nucleus. SRABP is now free to go to the nucleus and go and act as a transcription factor for these de novo lipogenic genes, and that's why we're seeing this increase. And so this is our what we're thinking so far is happening, at least in our dioxin-like female livers. So we also had some really interesting findings where we saw that our estrogen receptor alpha expression was going down in these livers. We're not going to talk about the AHR-ER alpha interaction today for the sake of time, but I'm going to show you another receptor-receptor interaction that we think is at play in these mice. And so what about estrogen signaling, right? That is one of the primary things that really, at least for the liver, helps, makes a impactful or instrumental role in shaping those sex differences or sexual dimorphism. So there's been a paper that came out from the Nagishi group in 2020. They're a great group from the National Institute of Environmental Health Sciences. And they were looking at these interactions between a constitutive androgen receptor, or CAR, with estrogen receptor alpha. And they showed that in the presence of diabetes or even phenobarbital, which is a CAR ligand, CAR gets activated, translocates to the nucleus, and through a series of conformational changes and binding with estrogen receptor alpha and subsequent phosphorylation, it can sit in this promoter region of this gene known as SALT21. And so this is an estrogen sulfotransferase gene. When it's turned on, it helps metabolize circulating active estrogen and activates it. So we were like, okay, let's go look at this pathway in our animals. So we did. So what did we see? So first of all, we look at hepatic CYP2B10, which is a target for the constitutive androgen receptor, to make sure that it's actually activated with exposure. And this is in our low-fat diet mice. And we saw that CAR activation seems to be different in our males and female exposed groups. So if you look here, there is a difference in the amount of CYP2B10 that was induced. And so we then look at SALT21. We said, okay, we see CAR activation. Are we going to see a change in a SALT21 like this, picture is demonstrating? So we did see that only in our female exposed mice, hepatic SALT21 was upregulated, but not in our males. So we thought, we were really excited about this, and we went ahead and said, okay, let's measure circulating estradiol levels and see if, it's really effective and metabolizing or inactivating circulating estrogen. And sure enough, when we look at our males versus our female exposed, there was a decrease in circulating estradiol levels in our females compared to our males. And surprisingly, we actually saw that in our males, plasma testosterone levels was actually up, indicating that perhaps this chemical pop mixture was causing this pro-androgenic, anti-estrogenic effect, and that pro-androgenic effect perhaps is driving some of those metabolic changes we're seeing in our males in terms of glucose intolerance. But the lack of circulating estrogen and estrogen receptor alpha interaction maybe play a role in our compromised metabolic dysfunction in our females. And so we were like, okay, we think we found a mechanism. What if we remove this estrogen signaling process? Will we still see that those effects it's going to confer any protection? So we then hypothesized that these POP mixture activate these xenobiotic receptors, impact estrogen signaling, and cause a series of effects to happen that eventually impact steatosis, glucose uptake, and inflammation. And so we did an overreactomy model to our mice. And what did we find? So this is a 2-week study. We haven't done a 12-week study yet with these OVX mice. But what we saw was that, and these are again, the same POP mixture that we exposed them to, we saw that in our males, these are all females. So we saw that in our sham operated, there was still that increase in area under the curve, showing that there was a intolerance with glucose uptakes in our females with exposure, but not in our not in our OVX mice. And we saw the same trend with liver weight. It was upregulated with exposure in our shams, but not in our OVX. Kind of confirming that perhaps that mechanism we thought to be at play was removed, and that's why we're seeing these no effects in our OVX mice. And we saw that with fat composition, for instance, OVX mice tend to have more fat, and that's something that's been shown in literature. But we also surprisingly saw that hepatic cholesterol level was upregulated in our OVX exposed mice. So this goes to show that with research, not everything aligns, perfectly. And sometimes you do get things that you still have to figure out why is that happening. And that's just an example. All right. So what about the gut? So I told you we look at the endocrine gut liver axis, and I'm going to show you just a little bit of study that we did with the gut, a snapshot of it. So this is another exposure study where we look only at PCB mixtures. There were no organic chlorine pesticides in this mixture, and there were just PCB 126, which is a dioxin-like PCB, and Aerochlor 1260, which is one of those heavily chlorinated PCBs that are still present in fish, right? So we are still, consuming fish products. We're still getting Aerochlor 1260 in our bodies. And so we did this was a 2-week study, and we decided to look at the gut microbiome. So this is a picture of beta diversity, looking at bacterial differences between the different groups. These are male and female mice, not exposed and exposed. And you could see that in terms of bacterial composition, they're all different. So all 4 groups are different. And we also look at the differences in the kind of bacteria that there are between these male and female mice with and without exposure. And I'm going to show you just one figure that I thought was interesting. So this was in the male controls versus male PCBs at the genus level. And we saw that there was this Dehalobacterium genus that was upregulated in our male exposed mice, but we didn't see this in our female exposed. So why did this jump out at us? Well, the Dehalobacteria family or Dehalococcoids genus of bacteria are they used oxidation of hydrogen for their respiration. So it's called halide respiration. And in doing that, they subsequently lead to dehalogenation of halogenated organic compounds. And this halide respiration has actually been used substantially in terms of PCB remediation or even other kind of organic compound remediation from soil and water samples. And we were really curious to know why this is only upregulated in our males. Could this be because of these chemical distribution in the body being different between males and females? Are they more likely to be metabolized or by these bacteria in our males? We don't know that yet, but it's really exciting to see this microbial signature being different and pointing out an exposure effect. And other things we saw with our gut, we looked at allele gene expressions. We saw that our female exposed mice had a lower expression of some of the permeability markers. And again, these mice, we already looked at the hepatic, indices for these animals, and we actually saw that again, our female mice, similar with the other exposures, tend to show more steatosis and more inflammation than our males. And we also look at the AHR in our intestine, in the intestine of these mice. And surprisingly, we saw that the CYP1A1 was induced only in our male exposed, but not necessarily to an extent to our females, but not as significant as our males, suggesting perhaps that there is this difference in intestinal AHR activation. And I want to point out here that we have looked at both hepatic and intestinal AHR activation in these animals. In our males, we saw that there was, a massive AHR, CYP1A1, CYP1A2 induction in our females. We did proteomic analysis on these animals, and all our computational analysis also kept telling us that there was an enhanced AHR activation in our females in terms of the liver. But it was really unique for us to see that there was this sex-specific differences in intestinal AHR activation. And this is important because for those of you that know a little bit about AHR biology, we know that the intestinal AHR plays actually a very beneficial role in terms of enhancing gut permeability and could be used as a potential intervention strategy. So I think I'm not going to show you more data on this, but this is one of the key data that kind of have been hinting at us that perhaps we should pursue looking at the intestine a little bit more and even modulating these differences in AHR activation between the sexes to see if we could manipulate this as an intervention strategy. All right, so with that, I'm going to stop showing you data, but I'm going to talk a little bit about our ongoing research. So what I've showed you with those exposure studies was, this concurrent feeding of high-fat or Western diet with chemical exposure. But really, as humans, we are exposed to chemicals, unfortunately, since the day we were born, even in utero, right? So these chemicals act as this first hit to our body and even to our livers that could lead to the compromised liver state. So it may, cause the liver to be slightly injured, but not really a clear manifestation of steatotic liver disease, if you will, but it's compromising it. And then as we grow, as we keep getting exposed, and as we, make personal choices about our lifestyles, even, we're introduced to other factors like a high-fat diet or even alcohol consumption or even smoking. And that could act as a second hit or even a multi-hit that could then push that compromised liver and make it a full-blown mast cells or a full-blown tassels, if you will. And so with that in mind, with this hypothesis, we now are going to start looking at a different kind of exposure paradigm. So what we're going to do is we're going to prolong the study duration because we are interested in looking at exposure with time. We're first going to expose these animals to this chemical before introducing a diet component. So we are excited to pursue these studies, pending we get funded for it. And then other studies that are ongoing or we plan to do in the future, also looking at humanized models. We know that there are species differences in the aryl hydrocarbon receptor. So I know that there are a lot of AHR skeptics out there. They're like, great, you're seeing these effects in mice. What does it mean for human exposure, knowing that there are these species differences? So we're hoping to use humanized models to answer some of those questions. We want to see if estradiol supplementation would help our females with that steatosis that they're manifesting. We also want to look at pancreatic toxicity because we're seeing these changes in glucose levels and wonder if there is direct pancreatic toxicity that's impacting insulin production, or even if those glucose levels have any impact on glucose-stimulated insulin secretion. And we also are interested in looking into potential therapeutic and intervention strategies. And so with that, I just want to, my closing argument. I've been watching too much Kamala Harris campaigns. My concluding remarks. So, I was telling you about that narrative that we have, right, about how males are more at risk for liver disease than females. But we now are taking chemical exposure into account, and our data are telling us otherwise, that perhaps females seem to be more at risk. And these were early adult male and female mice. And so our findings are actually questioning this narrative and perhaps changing or challenging this paradigm that, this may not actually be true. And the more research we do, the more we'll know about it. And we're also interested in looking at how these chemicals affect these different life stages, taking into consideration disease origins, developmental origins of health and disease, and understanding that especially with females, as we grow older, as we age, there are these changes in hormonal landscapes and how that or chemical exposure impact that and vice versa. And so with that, I want to thank everybody that's involved in my research. First of all, I want to thank my lab. They're not here today because they're doing a glucose tolerance test on our mice. But I want to thank them for all they do. They are the ones that generate all those data and helps with, all the day-to-day activities in the lab. I want to thank our environmental liver disease group. I see Bert Watson here. Thank you for showing up and supporting me. I also want to thank I have a KO1 NIHS award, and I have wonderful mentors for that award, including Dr. Carrie Kling from biochemistry, Irina Kirpich from our division, our group, liver disease group, as well as Irini Potnagar. My collaborators, some of them are here today. I also want to thank all the centers that have been supporting and really doing a fantastic job of supporting early stage investigators. And importantly, I want to thank the Department of Medicine for always being supportive. I get asked this question a lot. You're a basic science researcher. Why are you still smiling when you're still in the clinical department? And part of that is because of, they support research like no other. And these research grant rounds are an attestation to that. So thank you so much. And I will be happy to take any questions. Thank you.
So, I just have one quick one. So in humans, one of the biggest differences is percent body fat. And so if the toxins are lipophilic, they go into the adipose tissue. And then is the serum level higher because there's this constant equilibrium from this high concentration? Is that part of the mechanism?
That's a really good question. Yes, we think that that is one reason, especially why we're seeing those circulating levels in our females. It could be that because our males tend to have higher body weights, higher body size as well. But it also depends on we've done these studies adjusting for BMI, but we're still seeing that. So it could partially explain for it, but I think there are also differences in metabolism of these chemicals even that could drive, play a role. So you still have metabolites of these chemicals circulating, but not necessarily the parent chemical.
Sure.
Yeah.
Yeah, congratulations on your presentation. I have a question because you focus on estrogens mostly, but women are under influence of of follicular stimuli. And all these hormones, and especially in older women, follicular stimulin is still very high because wants to activate ovaries. It was first question. How about not only hormones from ovaries but from pituitary? Yeah?
Yes.
And second question, what is very important in all these damages, liver regeneration plays very important role. And liver regeneration occurs by dividing of hepatocytes or is going step far earlier from oval stem cells in the liver, which are around Herring ducts. Is any data how sex hormones affect this oval stem cell population in the liver and potentially, regeneration?
There's no data yet so far, at least in the context of chemical exposure to look into that. And in terms of how that hormonal landscape would age. So there has been some studies that have looked at. Unfortunately, with mice, they don't go through menopause. It's kind of hard to kind of reflect that with humans, how humans behave with these exposures. But one of our studies with the dioxin compounds is actually to do that. We're going to expose the mice, just let them hang out for 42 or 50 weeks, and then we're going to look at those parameters. So, but you actually just gave me a research idea. Thank you.
Okay. That's what this conference is all about. Okay. Yes, sir.
Yeah, part of my question is what Dr. Adachek asked you, because I work on pituitary hormone and reproductive hormone. So you showed some PCB effect on estrogen, testosterone, but what is the end result on reproduction, on fertility in case of female, or spermatogenesis or sperm count in case of male when they're exposed to chemical or PCB especially?
Quick question. So there have been populations in the world that have had they went through these accidental exposures because they were consuming. For instance, there was this Taiwanese population that was consuming the contaminated oil that had PCBs and PCDFs, and they've looked at those populations. So with those really high levels of exposures, there are reproductive effects, right? There was low birth weights that resulted from it. There were stillbirths and miscarriages that have resulted from those. So, but those are really high levels that generally, you and I are not exposed to. So we don't know in terms of with the low level exposure. So when we, that's why we're interested in looking at that, the effects through lifetime and then taking those biological elements that occur through the life stage of a woman to kind of see those effects.
Yeah, because that's a important question, because estrogen level may be up or down, testosterone, but it's not affecting the end product, which is the birth rate or pre-birth or whatever, abortion or something, then.
Yeah. No, that PCBs are known, even dioxins are known to have really massive, impactful reproductive, health outcomes. Again, we're interested in looking at our environmental exposures, so low-level exposures. Yeah. Any more questions?
It's very intriguing that your data suggests there's an interaction between the AHR and SREBP. And that suggests a lot of potential mechanisms. SREBP is regulated at the transcriptional level, but it's also made as a precursor protein that's retained in the ER and two specific proteases activated. So where in all that do you think the AHR interaction is occurring?
So I think it's interacting with the precursor, and we didn't come up. There was another study that kind of looked or suggested that, and that's when we, followed up on it with our own study. But I believe it's happening with the precursor, and then, holding it back in the cytosol for that reason. And then once, and that's why we hypothesize that once the AHR is activated and it's, degrading, then that's just frees up the SRBP to do what it generally needs to do. To activate lipogenesis. And the fact that it's more pronounced in our female exposed, we think that that is a mechanism. I don't know if that answers your question, but I believe from what I remember, it's the precursor.
Okay.
Yeah, we haven't looked at our protein levels for SRBP nor for cytosolic versus nuclear AHR, but we're doing that. Yeah.
All right.
All right.
Don't see any, thank you very much.
No, really?
What do you think we do? Yeah.
Okay. Right.
Should I leave the meeting?
So I.
Should I leave the meeting?
Oh, no, it's okay. I think it is.
Just put it on your first slide if you can.
All right.
Planning to teach them that. We need to have to leave in about